Hello and welcome to BIOS Frontier Science. I'm Kate Shannon, and today I'm excited to welcome Hani Goodarzi, Professor of Biochemistry and Biophysics at the University of California, San Francisco. To help host this episode, I'm joined by my colleague, Drew Yashar. To kick things off, Hani, can you please share a brief intro with us?
Sure. I'm Hani Goodarzi. I'm a faculty at UCSF and Department of Biochemistry and Biophysics, and also urology. I'm also a CZ BioHub investigator. I have a number of affiliations, including with the Helen Diller Family Comprehensive Cancer Center, Baker Computational Health Sciences Institute, as well as Baker Aging Research Institute and Parker Institute for Cancer Immunotherapy. Before joining UCSF, I did my graduate work in computational and quantitative biology at Princeton with my mentor, Saeed Tavazoie, who's now at Columbia, actually. And I did my postdoc with his brother, Sohail Tavazoie at Rockefeller in cancer systems biology. And I started my lab here at UCSF in May 2016, so it's been 6 years.
Awesome. And before we dive into specific projects that you're working on in your lab, one question we like to ask our guests is, throughout your career, what has been your North Star or the common thread that has tied your work together?
I think basically how to balance mandates, right? So basically how to, and really needing to balance execution and exploration and research with education, basically how to walk this tightrope of opposing mandates.
Great. And to kick things off, start diving into specific things that your lab is working on, I'll hand it off to Drew.
Wonderful. Great to have you on the show, Hani. So excited to dive into the specifics here. One thing we want to kick off, one of the major efforts in your lab is approaching metastatic progression and neurodegenerative diseases is the use of both experimental and computational technologies, including oncRNAs, tRNA fragments, and much more. So starting with your work on understanding human diseases at this molecular level, can you give us an overview of your work?
Sure. I mean, even really beyond diseases, the fundamental question that my career in research is really built on is this. The idea is that every cell in our body, for the most part, has the same genetic blueprint. So the genetics is identical. So really, in the diversity that we see in cellular form and function, both in the in the context of health but also disease, is really how this genetic blueprint is interpreted. So this idea that which genes are expressed, to what extent, in what context, is really fundamentally the driver of complexity in organisms. And my job has always been, how can we actually understand the molecular basis of cell identity and how that emerges. Again, it can be in the context of health or disease. The idea being that if you have a cell that resides in state A and another cell that resides in state B, and basically you're seeing a transition from A to B, what kind of data do you need to collect from both of these states to be able to reverse engineer the identity of the regulatory programs or regulatory networks in the cell that are shepherding the cell from one state to another? Now, states A and B can be within the context of just healthy differentiation or developmental processes, or it can be in the context of disease, going from a poorly metastatic state to a highly metastatic state, or from normal tissue to a cancerous tissue. So it's really the understanding the basis of these transitions and the molecular basis of these transitions and the regulatory networks and programs that underlie them is really the basis of my research.
Incredibly insightful, great overview. I really appreciate it. And so diving in, I mean, specifically, computational technology is an incredibly hot area, already beginning to generate so much impact within industry and within research labs across the world. As we gain a better understanding of how to tackle these complex phenotypes, how will we continue to improve drug development?
So I would want to kind of really take a step back and put in context really what the role of computation in life sciences has been. I think, as you mentioned, it has been growing and it will continue to grow for the time being, I think. And a lot of this growth really comes out of this notion that life sciences has changed. we have been more and more over the past couple of decades, thanks to advances in technology, we have been really gathering large-scale, high-dimensional data from biological system. And really the advent of second-generation sequencing technologies has enabled much of this progress. And really before this progress, you had a group of quote-unquote computational biologists, but they were really theoretical biologists, mathematical biologists that were really doing broadly theoretical work, that there wasn't really much of crosstalk between what was happening and the experimental and the rest of the field, which is mostly experimental. It was really these advances in technologies, genomic and post-genomic technologies, that really brought forth the power and the need for computation and really making sense of this high dimensional data. And that has really gave rise to computational biology and bioinformatics as really vibrant and exciting fields of research on their own. But ultimately, I think that the challenge has always been and continues to be how these two ways of thinking can really come together, and how do you actually function in that interface. The reality is that folks who are very computationally strong don't necessarily know what kind of biological questions are out there and need to be answered. So as a result of that, you have large swaths of computational biologists working on very similar problems that there's really no need for it. There's diminishing returns, basically. I think protein folding is a good example. Right? As important as it is a problem, you really don't need as many people as they're working on it. It's just a very famous problem. So anyone from a computational background who starts to think about biology, that problem is the first one they see and they start working on it. Whereas like one layer deeper, there are many, many, many questions that need computational power and computational solutions in biology that are just not as famous. And you would just only know them if you're more of an insider in the biology field. The opposite side is also completely true. You can generate data from really exciting model systems that are experimental, and you wouldn't really know what is even possible in terms of computational models that you can build on this, or computational analytics that you can run on it. You do just what you can and what you're aware of. Now collaborations form around bringing these two worldviews together. It's not an entirely efficient process. So ultimately, I think as the number of multidisciplinary researchers grows, folks who are fairly comfortable on both sides, they can glue the two sides of this process together, and more have like crosstalk. And I think that is where we see synergies, like really strong synergies, that a lot of problems on either side can be solved. And I myself, having done this for as long as I have, I really don't think in wet or dry terms anymore. I just think in terms of problems, and I think about solutions. Sometimes those solutions are dry, sometimes they're wet, and really we don't really have purely wet or dry projects in our lab. It's always a problem that we are trying to solve, and depending on the step that we are trying to solve, one of the two tends to be the answer.
Yeah, that's amazing. I love this take a step back, look at the holistic approach of what this research can do, at least take impact beyond the tried and true path. And so just taking a step forward from your own research, outside of oncology and neurodegenerative diseases, where else could you apply these learnings from these projects to improve patient outcome?
I think, ultimately, even if you're a translational researcher like myself, in the end of the day, you really have to understand that an important point of, discovery science is really to allow for, the unknown unknowns. That really understanding that we do not solve a lot of these problems by really studying them. The entire field of research and science needs to be elevated, and allow for that elevation to kind of synergize and give you answers in different areas that you want. Even in, if you take oncology, for example, we have made some progress over the past 3 decades since really the first war on cancer, in the 80s. We have made a lot of progress in terms of dealing with some cancers, but a lot of this progress actually has nothing to do directly as the first step with physician scientists working on clinical trials. A lot of it comes from genomic technologies that have changed our understanding of what even means to have a different types of cancer and what to do about it. Like targeted therapies really come out of that understanding. Or even like public health measures have had a huge role in this that, again, is not something that you think about when you think about research around in the space of oncology. So it's really allowing room for, I mean, now we understand it better and better since something like CRISPR really comes out of bacteria, for example, right? So I think people have appreciation for it more and more, but the reality is that this has been the case for the long haul, that you can't really solve a problem like cancer, but really pouring a lot of money into it and just having people understand that one process. That's just not how it works. So that at every level we should keep an eye on that, and even within one lab and one group, you have to allow for taking steps back and really doing discovery research that doesn't necessarily have anything to do with the problems, the translational problems you're trying to solve, and which in our cases tend to be mostly human disease. So that's really fundamentally how I really think about it. And the problem, the way that I outlined it in the beginning, again, I think is very generalizable, and it's not really just about human disease. It can be really applied to any context, or even any biological system for that matter.
Yeah, 100%. Super insightful. I think that little touch in oncology brings us into our next topic. So I'm going to transition a bit into our second topic, which is evolutionary elements of cancer progression. So diving into some of your research, your lab has discovered orphan non-coding RNAs, or oncRNAs, that have been able to demonstrate that cancer cells can adopt these oncRNAs and carry out new regulatory functions, which is phenomenal. Can you speak more on this work and why it's relevant to metastatic progression in multiple cancers?
Absolutely. So, the way that we approach this, I mean, we didn't really set out to necessarily find oncRNAs, if that makes sense. Again, like it's a discovery space, right? The fundamental question that we were trying to answer was this. As I mentioned, much of my research has been centered on how cancer cells hijack different components of our existing regulatory programs to change the expression of genes in directions that would help them survive and grow. I mean, I'm anthropomorphizing it a little bit, but that's basically what it is. Now, we truly understand cancer to be an evolutionary disease, an evolutionary process. Going from one transformed cell to a full-blown metastatic disease takes a long time, sometimes even decades, and through many, many iterations, many, many generations. And cancer cells have to overcome, and a tumor has to overcome a lot of barriers that the body throws at it to actually get there. Cancer cells are the heroes of this story. The way that I'm telling it, of course, is the opposite, is what we want to happen. Now, the question that we set out to ask was, understanding how long this process takes, can cancer cells actually engineer their own regulatory programs and do things that do not really even exist in healthy cells for them to hijack? So this search for quote-unquote cancer engineered regulatory programs was one of the tiers of what we were trying to do when we started our lab here at UCSF, along with my amazing postdoc, Dr. Lisa Fish. This is one of the first projects that we set out to do, and we decided to, for a number of reasons that become technical, I don't want to get into it, we decided to focus on small non-coding RNAs as a space to look for macromolecules that have regulatory potential in them. And the easiest way of figuring out a way that this doesn't happen in healthy cells is to actually look for things that don't even exist in healthy cells. So we first asked, can we find the small non-coding RNAs that are not even expressed in healthy cells, as far as we can tell? So they are quote-unquote cancer emergent. And we already had some evidence of these RNAs already existing, so we set out to systematically search for them and annotate them. And these are the RNAs, and it turned out that like hundreds, if not thousands of them exist, and annotating them into a descriptive class is basically what we call orphan non-coding RNAs or oncRNAs. But that oncRNAs are basically RNAs that are cancer specific or cancer emergent, that are basically not expressed in healthy tissues, but they appear in cancer cells and are part of the cancer cell identity.
Phenomenal. And just getting a little deeper, as you're touching on potential, in what indications would these types of therapies be relevant? And how can this research really improve on existing options?
We have been thinking about this a couple of ways. I mean, as I said, we have annotated thousands of these oncRNAs. In reality, most of them are just along for the ride and most likely don't do anything. So it's really about actually prioritizing and finding the ones that are relevant for the disease, out of the ones that are quote unquote co-opted by cancer cells for relevant functions. And we have so far, at least in one case, we have done kind of a deep dive into how mechanistically this can actually happen, and we are working on other ones as well. This is useful from my perspective, in addition to really just exciting biology and really understanding the complexity of cancer. It is also the fact that these are cancer engineered pathways. So if they are really relevant and drivers of disease progression, they provide really unique opportunities for targeting them because they provide really nice therapeutic windows. Therapeutic window is basically like, the dose at which that you can basically provide a drug to inhibit a pathway that hits the cancer, but not really the healthy tissues. Basically, that's the best way to think about it, because any cancer, any drug can have on-target or off-target toxicity. Assuming that there isn't any off-target toxicity, there's also on-target toxicity. That, for example, you're trying to hit a cancer cell, but you can also hit other cells in the body, including the liver cells or heart cells and so on and so forth. So since these pathways are supposedly only present and active and in cancer cells, they provide little opportunity for on-target toxicity, right? So if you hit them, they would, like systemically with a drug, they should really be only affecting cancer cells. And I think that is like one way to really think about it. And also another approach to really think about it is that I think we are kind of only scratching the surface of cancer engineered regulatory programs. We just looked at small non-coding RNAs for, as I mentioned, as a place to start searching for these. But of course, there are many other modules and regulatory modules that you can think of that can come from other sources that are non-small RNAs, basically. And so they can play other roles that could be interesting and could augment our understanding of how cancer actually progresses, and offer other opportunities that to therapeutically intervene as well.
Amazing. And so as you bring this new approach forward, what do you believe is the biggest challenge, or challenges for that matter, will be?
I mean, I would say scalable discovery and annotation of these cancer-specific macromolecules is an important step in all of this. Especially since when you're talking about cancer emergent, then you have to go back and kind of talk about its non-existence in other cells and healthy cells for that matter. And proving a negative is impossible, right? So we keep looking to really filter out things that are present in quote-unquote healthy cells, right, to really rule out their function. So making that really scalable and having the access to kind of data that allows you to do that, I think, is certainly one barrier that we need to overcome. The second side of it is also kind of like a scalable annotation, like functional annotation. As I said, you detect things, but do they actually do anything? And that's also true for even genes. As we have about 20,000 genes. There are a large number of them we actually don't know what they do, and for the ones that we do what they do, we know them, maybe one aspect of what they do, and there are other aspects that we have no ideas about. So even something like established annotated genes that we have spent so much time with them, There's still a lot that we don't know, and we haven't actually functionally validated them. So think about this other space that suddenly arises that only our lab is looking for, and suddenly there is this massive space that you want to functionally annotate. And of course, we have a head start because now we have technologies that allow for kind of like high throughput parallel measurements. I think functional genomics has had provided us with openings to be able to do these kinds of parallel functional annotation of many players in the cell, and we are taking advantage of these resources in similar fashions to study this quote-unquote cancer emergent potential putative regulators.
Right. And so now, taking the problems, seeing the potential of this new research, would love to just take a step back before we dive into the next topic. How do you really see the field of oncology changing over the next 10 years with this technology?
I think there are a few answers to that question. What really comes to mind, and I'm sure everyone would talk about, is precision oncology, really appreciating the individuality of every tumor and how you need to approach it. But I would say I think our biggest challenge, very likely in the upcoming decade, is really disparity of care when it comes to cancer research. I think those disparities will continue to grow if we don't pay attention and we don't do anything about it. So I think that's really one challenge that we are aware of, and the field as a whole is actually working on. But we certainly can spend more resources on it. I think fundamentally, the bottom line is that when you talk about cancer, and everyone understands that earliest stage cancer is easier to deal with than later stage cancers, right? So fundamentally, in everyone's mind, cancer is a diagnostic problem, right? So if you really had the tools to identify and detect cancers, then you're good to go, right? Then you don't really have to deal with treating late-stage cancers, which is very difficult. It seems to be a losing battle. The reality of it is that actually detecting cancers that you have to do something about is not trivial. Detection is two sides of the the same coin, right? And it can lead to overdiagnosis. The reality is that cancer is a very strongly age-associated disease, and almost every old person has tumors. A lot of these tumors are not going to do anything. They are slow growing, and they're not really aggressive, so they're ultimately not going to be the cause of death. But now, if you actually have a device or instrument or approach that has 100% sensitivity, suddenly everyone, above 65 or 70 will have cancers. And so we really need to define what are quote-unquote high-risk cancers, like what are actionable steps that we need to take. What is about the biology of that cancer that makes it a worrisome one that we actually need to take steps. So we need to basically internalize rules around risk, that what kind of cancers do we actually need to actively work on, and which ones should we just step back, because every action and intervention has a cost to the patient and actually impacts their quality of life. For example, in prostate cancer, active surveillance is becoming more and more common, and I actually believe it started out coming out of UCSF as one of the centers that really promoted active surveillance as an approach for prostate cancer. So I think that is actually another direction that we really need to think about, that which fights in cancer are actually worth fighting and really can impact patients' quality of life.
Yeah, incredibly insightful. Great overview on cancer the next 10 years. And with that said, would love to move on to our third topic, Hani.
Sure.
Covering post-transcriptional regulators in neurodegenerative diseases in your work in that field. So in addition to your computational research in oncology, your lab is also working with the Tavazoie labs as well as the Tavazoie labs to develop computational frameworks within neuronal development and neurodegenerative diseases. just to begin with, can you just give us an intro into your work here?
Absolutely. I mean, as I mentioned, the question that we work on is fundamentally more generalizable than really what happens in cancer. And so, of course, we develop tools and approaches and systems to study these kind of like state transitions that I talked about. And in the context of our own lab, we tend to collect data from, I mean, cancer is our model, right? So cancer progression is specifically in metastasis. So that is the kind of experimental model that we have built. We can collect data, we can do validation. So that's the system that we are active in. But the platform we have built is more generalizable and can be applied in other contexts as well. So over the years, we have done that through collaborations, and the Tavazoie Lab and Rockefeller Labs are some examples, but there are other ones as well, especially here at UCSF that in the neurodegeneration space, but also other diseases or even non-disease systems that where we bring the know-how and technology and the approach that we have built to study, cancer progression, but really apply them to any kind of progression, be it healthy or disease, and use it to really dissect, again, the regulatory programs that are giving rise to these progressive changes.
100%. And when we talk about neurodegenerative diseases, some without good levels of care or pathways for cure yet, one thing that comes up is Alzheimer's disease. So would love to dive in and see what is the relevance of this work for Alzheimer's, among other neurodegenerative diseases that are in need for considerable new therapeutic targets. Would love to dive in and just see how that work fits in.
Absolutely. I mean, that is one of the questions that we have ourselves. So partly to figure out the functional relevance of these pathways. But I would preface this by saying that RNA binding proteins have long been implicated in neurodegeneration, which RNA binding proteins are master regulators of post-transcriptional gene expression control, and there are actually genetic variants that have been known for quite some time that promote neurodegeneration. Now, the nature of the role in disease is really not fully understood, and it's likely very complex and multifaceted. Because whenever you have like a variation, you can think of it both in terms of loss of function and gain of function. So what are the functions that a given RNA binding protein is not playing in a context of disease? Or what are the pathologic functions that it's now playing in the context of a disease? So RNA binding proteins tend to be these very multifunctional proteins. That's partly why they're so exciting, but also very difficult to study. The reality is that we have think of like a few hundred RNA binding proteins are around, and they regulate every aspect of RNA life cycle in the cell. So from the moment that the RNA is transcribed from DNA, you're within their purview until like you're degraded, right, as an RNA. So basically, all of that is controlled by these proteins in ways that is very complex and combinatorial, and really we don't understand the grammar of it even within healthy cells, let alone how these are disrupted in the context of diseases such as complex diseases such as cancer and neurodegeneration.
Amazing. And taking a step back here, again, as we're getting to the frontier and the fringes of science and research, looking forward and ahead, as we continue to pursue neurodegenerative diseases, what are some of the technologies that you are most excited by that you're seeing in this space currently?
I mean, I would say, kind of like as an outsider to neurodegeneration, I've always been neurodegeneration adjacent. Most of what I've done in that field has been through collaborations, really going back over a decade. I've always just collaborated with folks who have been working on this. And in contrast to cancer, I would say one of the biggest challenges in neurodegeneration is really having reliable disease models. And depends on your definition of disease model, and it depends on your definition of reliable. But for example, in cancer, you have cancer cell lines grown on a dish, which is not really great models, all the way to patient-derived xenografts grown in mice. So that's kind of a range of diseases. None of it is really a perfect recapitulation of what happens in a human patient, but they, depending on the question you're asking, they can provide levels of insights towards that. But the extent to which those levels are, those insights are generalizable and relevant to human pathology is ultimately the question that we as scientists actually care about. Now, in the space of neurodegeneration, you have models that some of them are new, some of them are older. Cultured iPSC-derived neurons, for example, is one way, or brain organoids, newer ways that people have been modeling this in culture, or even like genetic mouse models that are really commonly used as experimental models for studying this process. But the reality is that neurodegeneration is very complex. Neurodegenerative diseases are very complex. So these models lack different aspects of it. And partly, I think, as we make progress, better models are going to make this field more and more engaging. And the other side of it is our more recent abilities to collect really high quality, high dimensional data from patients. I mean, the reality of it is that one of the reasons that cancer has made as much progress as it has is like access to biopsies. Now and being able to process them and look at and collect data from them. And now the same also applies to neurodegeneration as well. But more importantly, what kind of data we can get from postmortem biopsies. Taking advantage of single cell genomic technologies that has really allowed us to look at both the spatial but also heterogeneity of different cell types in the environment and how they change in the context of disease, and basically get better data from human patients that we can help us put better in context what we see in models, either cell-based or mouse-based models.
Wonderful. Thank you so much again, Hani, for diving into your work and a little bit deeper into the ecosystem itself. I'm going to pass it off to Cage now to jump into our next topic. Cage.
Thanks, Drew. So, Hani, in addition to the incredible work going on in your lab, you also recently launched Exile Bio to bring forward some of these discoveries in neurodegenerative diseases and now more recently in oncology. Can you give us a brief introduction into Exile?
Absolutely. I mean, I described what led us to orphan non-coding RNAs or oncRNAs. As I said, they are cancer emergent. Now, what is interesting is that as we were looking at this class of RNAs, at some point we actually realized that they're also secreted by cancer cells, like a fraction of them, and that became very quickly and very clearly important to us from a diagnostic perspective, because if you see oncRNAs outside the cell, since they're not really expressed in normal cells, then it means that there was an underlying cancer cell that released them. So if you collect biofluids from the body of a person, if you see enough of these oncRNAs, it means that there is a tumor somewhere that's actually releasing them. Now, as I mentioned, since there is a very strong tie-in between which oncRNAs are expressed in a given cell and the regulatory state of that cell, there is this much tighter association between oncRNAs that you see and the biology of the tumor, much more so than other modalities in the liquid biopsy space that people have been looking at, which is, for the most part, DNA-based modalities. Now, that is really the basis of the foundation of Exile, that you're looking at oncRNAs as an analyte for detection. That, first of all, is a digital presence and absence signature, which provides a lot more robustness than quantification-based signatures. Another part of it is that they have a very strong tie into the biology of the cancer cell, so they provide a window into the biology of the tumor, which again goes back to that idea of not only we need to be able to detect cancer, but we also need to be able to tell something about that. It's a kind of cancer that we should be worried about or not, right? So oncRNAs is potentially a modality that would allow us to do that. And also, since there are RNA molecules that are actively secreted by cancer cells, they just tend to have higher baseline abundance, we think, that will allow us to achieve higher sensitivity than other liquid biopsy modalities that we have seen. So that's certainly the premise that Exile is built on. the reality is that at some point, you see a framework and the promise of it, and then you have to build a team and trust in them that they will actually materialize that vision. And so that's certainly the vision. That the XI is built on being able to do what we call liquid pathology, that really from blood samples, small volumes of blood samples for that matter, be able to tell not only that there is a disease, but also tell something about the biology of that disease.
Awesome. So you mentioned before kind of the importance of being able to differentiate between a patient who has a tumor and a patient who has a tumor that might actually have some negative consequences for their health. Exactly. But can you take it a little step further and kind of explain how you see Exile changing the way we address oncology over the next 5 to 10 years?
Yeah, I mean that's exactly the point. That the biopsies in oncology space are useful because not only they confirm the presence of cancer, but they tell you what to do about that cancer. You'll have access to stains and data and morphology and like what pathologists basically look at to tell you like what stage you're looking at. So basically, how high risk is this, and also give you information about subtypes. Subtypes are important because it tells you how you fight them, right? So in our excitement and rush towards like translating biopsies to liquid biopsies, we kind of got stuck in the first step of it, which was detection. I mean, of course, it's a revolution, and that's really exciting. Again, everyone really views cancer as a diagnostic problem, but we forgot the latter part of it to some extent. That biopsies are useful not only because they detect the disease, but also they make it actionable, right? So that is, I think, where I'm excited as a scientist, and ability of OnCarNase to open a window towards that you can really tell all you need to know about, basically replace biopsies, right? So everything that you tell about like a solid tumor biopsy, you can now be able to tell just from a liquid tumor biopsies. And what is that useful? Why is that useful? Is because it's small volumes of blood, very non-invasive, so you can do this very longitudinally over time. And if you can actually keep an eye on yourself as someone, like as you grow older, that creating that baseline information over time can be super useful. And so you can know like when things change, and when things change, like in what direction they're changing, and whether that direction is actually something that you should be worried about.
Amazing. It'll be very exciting to watch that company progress over the next couple of years. But kind of in addition to your company, do you have any advice to young researchers who are seeking to translate their work from academia into the industry?
Yeah, absolutely. I mean, I touched on it a little bit. I think for us academics, ultimately, the question is, when is the right time for an idea to be spun out, and who's the right people to do it? And I think these are very non-trivial questions. Part of it, for Exile, for example, or any other platform, you get to a point that you have de-risked what you can do as a scientist in academia, right? Nothing much changes, if you put more time and effort and money into it. So that's one. So what I'm trying to say is, like, at some point it stops being science in some sense. It stops being academic science is a better way to say it. That, like, for example, it doesn't really add anything to a postdoctoral graduate student's career to do more of the same, right? If you have sequence like process 200 patient samples, does it add to sequence or process another 200? The answer is probably not. Right? So we are not in the business of commercializing and product development, right? We are academics. We are excited about the science, academic science side of it. So at some point, it's very clear that this has stopped being, academic, right? And that's certainly one way to tell that, this is, you need to spin this out. The second side of it is to really understand, we are academics. Our job is tough as it is. Everyone will tell you how much effort and time it takes to run a lab. So we certainly are not going to be able to run a company on the side. So you really have to find a team that that shares your vision of like how is this done, right? In what is the ideal form of realizing this platform or framework? And I don't think people appreciate how long it actually takes to find that team. So my suggestion is to start early, and even before you think you're done with the platform, if you saw the promise of it, and start talking to people, just reach out to your network and work with them over the years to basically make sure that when the time comes, there is a reliable team that who is as committed as you are to the vision that you have, because you're not the one who's going to be there and working on that vision. So you have to trust in their judgment and their ability to really do it, basically, and get it done. And with Exile, that's exactly what I had, and that is the model that I've also continue to recreate going forward as well.
Great. I think that's some really good and insightful advice for our listeners. Moving on and changing tack slightly to our last topic, your research in cancer biology recently led you to being awarded the 2022 Vilcek Prize, a prize dedicated for promising young immigrant scientists and artists in the United States whose work represents a profound advance in their respective fields. What does this award mean for you and for your lab?
Oh, it means the world to me. I've received other awards. I think part of being an academic scientist is that there are plenty of awards that go around. Like, we are very good at patting each other on the back with different awards. But this one was really different for me, partly because, in large part, because it's really devoted to immigrants. And being an immigrant scientist is part of my identity. I moved here back in 2006 for grad school, and that has really shaped the kind of scientist that I am. And I continue to work with friends and colleagues who are also academics that were classmates back in Iran. So partly because of that, I was really excited about this award, and also just broadly really understanding the work that Vilcek is doing. There are not many foundations that are largely devoting their efforts to promoting immigrants and immigration issues in general.
Awesome. And along those lines, is there more you think we can do to support and mentor first generation and immigrant scientists?
Oh, absolutely. No question. If anything, as I said, I moved here in 2006 from Iran. It's never been an easy time to immigrate from certain countries, and Iran is certainly one of those. But we have had good times and bad times, and more recently bad times. I think travel ban was a good example of that. But by and large, things haven't really changed for the better. For us to provide an opportunity for young minds and scientists to come and work and study in the U.S. And we don't really get to benefit from their presence. It's part of the equation as well. Not only we get to benefit them, but the opposite is true as well. If you look at the faculty here at UCSF, you get massive enrichment for immigrants. Some of whom actually, like me, moved here for higher education, and that's certainly something that we need to work on, kind of like as a nation, right? Because otherwise they'll end up other places, as they are. So we are creating an environment in which exciting science is done, but we also need to make sure that if we are bringing international students, we actually provide enough attention to them, realizing that a 20-year-old who is moving here, they're young, they don't have a support network anymore. They're basically supplanted from there to here. Some of them have visa issues, like I did. They can't go back and visit their family easily and at the drop of a hat. Some of them are stuck here for years without friends or relatives or any kind of financial or moral support. So we have to be cognizant of, like in academia or in general, as a society, cognizant of the burden that immigration actually, the toll that it takes, especially on young minds. And we have to be prepared to, A, help them overcome it, or if possible, just remove the burdens. I think like visa issues are just absurd for students to have. They should be able to go back and forth as they please. It's not that difficult of a problem to solve, but it has existed since I remember, and it's going to continue to exist. It's just because it impacts such a small minority of folks that no one really cares. To the extent that I can, have been vocal about it, and I know other international scholars have been vocal about it, but nothing much has changed.
Thank you for sharing that. Thank you for joining us. It's been fantastic to hear your experience as a researcher, entrepreneur, and as an immigrant. But before we come to a close, I want to ask you a few rapid-fire questions to cap things off. First, do you have any advice for early career individuals hoping to pioneer new fields?
Absolutely. I think going in a new direction that you think no one else has is of course risky. So you have to kind of like risk manage your portfolio of projects, the way that investors risk manage their portfolio of projects. So you need to be able to offset or hedge your bets. You're going to invest in a direction that you think is exciting and has a potential to really give rise to something completely new. But you need to also offset that with projects that are more within your wheelbarrow, and you're more confident and you can actually get this to work. So certainly don't put all your eggs in one basket. Don't shoot for the moon. It's important to pace yourself and understand that discovery takes time and effort, and you need to exist and for that sustained effort to reach fruition. So again, it's all about risk management.
I think that's some great advice. And do you have any other closing thoughts or shameless plugs you want to share with us before we go?
Yeah, I would say academia is pretty great. I think these days for some reason everyone thinks otherwise. This is not an easy job, not by a long shot. No one certainly is in it for the money or anything like that, but it has a lot of positives. Getting to shape the minds of the next generation is certainly part of it. There's a service component to the job for sure, but there's a lot of excitement. Our ability to really do curiosity-driven research is really amazing. I mean, being in an era that you get paid any amount of money to just follow your curiosity is really amazing. So you can always have more, but also you can take a step back and appreciate what we have. That we can have a cast of like professionals who just can devote their time and effort into following their day-to-day curiosities. Could it be easier? Should it be easier? Should we do more of it? Of course, there's no question, and I'll be there in the front lines with you arguing for that. But there's also this appreciation that all it takes for us to go in a new direction, to come up with a new idea and pursue it, is really like 2 of us getting together, sitting, drinking coffee and just brainstorming. And once we decide something should happen, by and large can happen. I understand that, we are also very privileged to be in a place at a time that we do have the resources to do that. But that kind of freedom you will seldom find elsewhere. And of course, there are other things that you have to do for that privilege.
Awesome. And for those who are interested, where can people, learn more about you and your work?
Academics are very visible. Just, search Google Hani and UCSF and you'll find me. I'm on Twitter. My lab website is fairly up to date, and my email is on it. Yeah.
Awesome. Well, thank you, Hani, for an absolutely incredible episode. I'm sure our listeners had as much fun as we did. We're incredibly grateful for your time.
My pleasure. Thanks for having me.
Thanks, Hani.
Have a good one.
Take care.
Bye.